Hello everyone. I want to welcome you to Hawthorne's webinar series. My name is Paula Bartholomew. I'm the Director of Student Services at Hawthorne, and it's my pleasure to be the moderator for this webinar. I'm here with James Bernardinelli, who's engineering this presentation, and with Dr. Janet Ludwig, who is our presenter. I want to welcome you all and thank you for joining us. We're just about to begin the presentation about the microbiome. It's new territory, and our presenter again is Dr. Janet Ludwig. And I want to give you a little bit of a description about this presentation, because we're going to talk about and answer a lot of questions during this. Dr. Ludwig will. And we're going to go into the microbiome and ask if it's the answer to many diseases that plague us today. It's certainly an interesting possibility that a fecal transplant from a healthy person to an ill person could bypass the use of antibiotics and other medications that have become commonly used in today's medicine. So what about the development of your microbiome? The question is, are you from a family that's prone towards weight gain? Or could it be that your microbiome predisposes you to being unable to lose weight? Also, environmental toxins and how they alter the genes of your microbiome could also account for health issues that might be affecting you. So during this presentation of the Microbiome New Territory, it's going to address these issues. And it's new and exciting information that will be explored to answer many questions about the microbiome, like what it is and how it can influence health. And similar to the earlier project of elucidating the human genome, this was completed in 2003. Now the Human Microbiome Project was launched in 2007, and already we're seeing results from these studies shedding new light on chronic diseases and many conditions that have not responded to traditional treatment. So of interest are the many laboratories and studies that are being done on individuals specifically to elucidate their unique variation of the microbiome and possible predisposition to diseases. That said, let me tell you a little bit more about Dr. Ludwig. As we've said, Dr. Ludwig is Hawthorne University's Education Director and Director of Doctoral Studies Program. Dr. Ludwig has worked in the area of inflammatory diseases for more than 25 years. She was at the Division of Surgical Biology at the Arizona Health Sciences Center studying zinc and cell injury, which was induced by alcohol and carbon tetrachloride, an effective hepatotoxin. And additionally, she has studied mechanisms and structural identification of the potent class of inflammatory mediators, specifically platelet activating factors. She did this at the Department of Pathology at the University of Texas Health Sciences Center in San Antonio. She's also the author of peer-reviewed articles and book chapters and has presented her findings at national symposia. She's been the Dean of Sciences at Nurse Anesthesia School and has taught courses in inflammation, biochemistry, and nutrition at various universities. And some really important work here. I just love this. Dr. Ludwig also travels to Bangkok, Thailand every year to give nutritional advice in a non-governmental organization that aids in improving the conditions for impoverished women and their children. And currently, but not lastly, she's on the Hawthorne University faculty in the doctoral studies program. So in addition to being education director, she's also teaching our students, which is a wonderful thing. So it's a pleasure to have Dr. Ludwig presenting this webinar. Welcome, Janet. We're so glad to have you with us. I know that you have a lot to cover, so I want to let you get this presentation underway.
Thank you very much, Paula. I appreciate the very nice introduction, and as an added to that, I really enjoy being a part of the Hawthorne University. And specifically to this talk, I am delighted to be able to present and share this exciting new information. As you'll see on this first slide, Microbiome New Territory, this is an image of the microbiome. In this case, these are E. coli that are within the villi in the gastrointestinal tract. And we'll be exploring all this exciting new information. Specifically, I will be answering the questions relative to the microbiome. What is it? That's certainly paramount to going on with the discussion. Why do we even care about it? What does it do for us? Can we change it? Profound studies are being done on this in this area. Next, we'll discuss how does it influence our health. This is a very new and exciting data that is being presented that I'll be going over today. Some of it, not all of it. It's a great deal of information. Where can a person have his or hers determined? There are certainly laboratories that are out there that will do the bacterial species of your microbiome. And lastly, can a person be a fecal transplant donor and recipient? We'll go over some of these and the pros and cons for these types of situations. There are certainly new exciting studies on microbiome. It has been present in the media. Just September 6, 2013, Dr. Jeffrey Gordon, whose studies I will be reviewing a great deal, he did a podcast on NPR, Do Your Gut Bacteria Influence Your Metabolism? Michael Pollan, of course we all know him, he did a report in New York Times, May 15, 2013. Then, of course, the recently published studies reporting success with fecal transplants to overcome resistant GI infections. This has certainly been top of the news. And here is the paper that shows success with modifying gut microbiome fecal transplants. Clostridium difficile is a particularly difficult GI infection. It does not often respond to antibiotics, and those are even expensive. There has been a great deal of success with using fecal transplants from healthy individuals, putting them into these individuals. Basically, they have reported 90% of the patients have been cured. So this is very exciting studies that can bypass the use of antibiotics in some of these resistant strain infections. So, to overview of my presentation, I will first be describing the microbiome. Secondly, we will discuss the diversity of the actual human gut flora. Thirdly, I will go through the development of the human gut flora from birth to adulthood. Next, a major portion of the presentation is the bacterial influence on the metabolism and obesity of the individual. Taking that information, we'll talk about altering the microbiome for health. We'll also identify labs where the individual microbiome can be elucidated. Conclusions in the future will finish up the presentation. So I'm very excited to go on with that. And the first portion is I will describe the microbiome. First of all, I wanted to give you a visual representation of the skin microbes. This child has, as we all do, we live in a world of microorganisms. And the dirt that is on this child represent the visual representation of the microorganisms on the skin. We have bacteria, fungi, viruses, not only on us, but also in us, which is what is of importance in this discussion. Here is a schematic representation, again, of microorganisms and the humans. And I want you to notice when we discuss this, be aware of the size of the circle. On the outside of the body, we have skin, a reasonable size of microorganisms. The number in the parentheses represents the phyla. As we go to the GI tract, we have in the mouth 56 phyla are present. And as we go down the GI tract, the esophagus We still have about the same relative size, but I do want you to notice that we have most of this is blue, representing the phyla Firmicutes. This will be important throughout our discussion. So we go through the esophagus, we get to the stomach. Again, we are getting blue, but we're also getting pink present too. Now we get to the very large circle, the microbiome, that we are primarily going to be discussing. And it's very large because of the number of microorganisms. It contains, notice, half of it Firmicutes and half of it Bacteroidetes. This is very important as we discuss the mechanism of the microbiome and how it behaves in the body. Taking this through the definition of the human microbiome, microbiome is the aggregate of all gut species. I pointed out the skin species and the vagina species in the previous slide, but we are only going to be discussing the gut species. And specifically, the term microbiota refers to the individual bacterial species within the biome. There are indeed over 100 trillion organisms, 10 to the 14th, that are residents in and on the humans. Over 500 species have been identified so far. 90% of the cells in our body are microbial. For every one skin cell, there are 9 microorganisms living on and in you. And of importance and profoundly, 100-fold more genes are in our gut than are in us. So we consist of a great deal of microorganisms. And our flora, the microbiome, they're an integral part of our genetic landscape and our evolution, as we will review in this presentation. As was mentioned in the introduction, the Human Microbiome Project to identify gene sequencing of the bacterial composition occurred in 2007. It's a 5-year project analogous to the human genome. Out of this, we have some very exciting studies that I'll be going through. At this point in my presentation, I now want to go over the diversity of the human gut flora. The residents that live in the anatomic regions of the gut, basically, we have in the upper GI tract 10 to the 2 to 10 to the 4th cells per milliliter. They're primarily lactobacilli, streptococci, and of course H. pylori, which causes ulcers. As we get on further, we have a proliferation of organisms. Now we have 10 to the 6 to 10 to the 12 cells per milliliter. And we have what are termed facultative anaerobes, those organisms that can live in the presence of oxygen as well as no oxygen present. We also have obligate anaerobes. These consist of our Bacteroides and our Clostridium species, some of which are pathogens. These organisms will die in the presence of oxygen. They are in a non-oxygen environment for survival. In the colon, the human colon is the most biodense natural ecosystem known. It has 10 to the 10th to the 10th to the 12th cells per milliliter. It's very complex and diverse. It comprises most of our bacterial biomass. I wanted to show this image because I think it's beautiful. This is H. pylori, Helicobacter pylori. This bacterium, as you know, is the main cause of gastric and duodenal ulcers as well as stomach cancers. It was discovered in 1979. It is gram-negative, flagellated, as you can see, the flagellum, helical, and indeed about half of the world's population is infected with this, even though they may not, these individuals may not exhibit these pathogenic conditions of the ulcers or stomach cancers. So now with this slide, out of the human genome, we do have identification of the human gut flora. Bear with me, there's a lot of information in here. As I talked about, before, we have phyla that are present. And most of our phyla, as I showed you in that original slide of the individual, about half of it are primarily Firmicutes. The other half is primarily Bacteroidetes. Within those, we have, under the Firmicutes, we have the Clostridium, which can be the Clostridium difficile. It's a very difficult GI pathogen. We also have the Lactobacillus. And then, of course, we have the H. pylori that is causing the ulcers. I have highlighted one other one because I will talk about that next in the development, the Bifidobacterium, that is important for infant development of the gut microbiome. So these are important. You'll see this slide again because it represents identifying the particular flora that are responsible for some of these actions, the mechanisms. I wanted to include this phylogenetic tree of life. We are, of course, eukaryotes. We're over on this side, so we are the host in this situation. Primarily, I'll be talking about the bacteria all the way over on this side, on the blue. The archaea is a small group that is in the phylogenetic tree of life. There do consist of organisms of the archaea. You'll notice that they are methane producers. They do regulate the metabolism. I won't be presenting that data today, though. So let's go on with the development of the human gut flora. Early gut colonization has 4 phases. First, of course, is the infant is born and has a sterile gut. Secondly, there is an initial acquisition through the vagina delivery, feces, and the hospital environment. Third, the infant will be either breastfed or bottle-fed, and depending upon the type of feeding, the microbiota is different. If it's the child is breastfed, there is more bifidobacteria. You may recognize this, that it is often used as a probiotic. If the baby is breastfed, then 90% of the flora is bifidobacteria. If the child is bottle-fed, then the microbiota is more diverse, more Bacteroides and Clostridial species are present. Phase 4 is the start of the solids, moving to the adult flora at around age 3 years. And if the infant was breastfed, bifidobacteria remain key into the adulthood, and the presence of that actually inhibits Candida albicans growth. There have been studies showing that result. So going back to this same slide, if the baby is breastfed, we'll have primarily Bifidobacterium present. If the baby is bottle-fed, there'll be Bacteroides and Clostridium present. So we'll see the difference in the distribution as far as the genus are concerned. Continuing on with the development of the microbiome, results from this studies indicated that there are oligosaccharides in human breast milk when infants do not have the ability to digest them. You'll recall that oligosaccharides are small-chain saccharides, mainly from 2 to 10 saccharides linked together. The gut bacterium, here's the name again, Bifidobacterium infantis. This is specifically nourished by this complex carbohydrate. So upon growth, the Bifidobacterium proliferates and crowds out the less savory bacterium and establishes a healthy epithelium for the infant. We, of course, know that a healthy epithelium of the intestines protects the infant from infection and inflammation. The conclusion, this data indicates that breastfeeding promotes a healthier microbiome in infants. Once the baby is on solid food, there will be continued maturation, and through adulthood there is increasing diversity of the flora as we age. The microbiota in serial stool samples of an individual, they show remarkable stability. The greatest degree of similarity occurs within twins. However, the host genetic influence has been unexplored. In other words, ethnicity, race, these studies have yet to be done. So what are the species in the microbiome? Summarizing, they're eukaryotic, bacterial, and archaeal. Primarily, there are two groups, Firmicutes, gram-positive, and Bacteroidetes. These two groups comprise over 90%. And within this, there are, as far as anaerobic to aerobic, there are 1,000 to 1. These are far fewer species that are present in the environment. There's a high level of interpersonal diversity, such that the bottom line is my stool is not the same as your stool. Continuing on with the presentation, I will now discuss the bacterial influence of the metabolism on metabolism and obesity. Gut flora and metabolism. Microbiota enhance our gut metabolic activity. You may do this indirectly or directly. The colon bacteria, they are very active metabolically. Basically, they metabolize 20 to 70 grams of carbohydrates, 5 to 20 grams of protein per day. This is over 100 kilocalories can be produced per day. If you're on a 2,000 kilocalorie diet, 5% is going to feed your microbiota. The mass of the colonic microbiome is equal to a signal kidney, and it's metabolically as active as the liver. Continuing on with this, there is an energy conservation producing short-chain fatty acids. These microbiota produce acetate, butyrate, and propionate. These are absorbed into the body and they're used by the liver and the other organs. 50 to 70% of the colonic cell energy is actually derived from butyrate. The number and the diversity of the organisms is key relative to this metabolism. And this begs the question, do we acquire flora that program us for obesity or for normal weight? Now this slide, lots of information, bear with me. The title is The Metabolic Reactions in the Gut Flora. Underneath of it I have put the subheading. It basically is the same reactions as your liver cytochrome P450 performs as far as detoxification. You'll see on the left side, it performs the gut. These are the gut microbiota. Hydrolytic reactions, reductive reactions, nitrisation, and removal of functional groups. Some interesting substrates are cellulobiose, which is a product of cellulose metabolism. Azo compounds are metabolized by these gut flora. And of course, DDT, the chlorine group, is removed. So very similar reactions are occurring by way of the gut flora, same as what detoxification occurs by our liver. Similarly, let's talk about the gut flora in the brain. In a normal situation, there is a gut-brain axis that is occurring. If it's a stable environment, then there will be stable microbiota and physiological or controlled inflammation. Upon stress, such that cortisol and epinephrine are produced, there will be an inflamed response, thus altering the bacterial composition, and this resulting in physiological inflammation that will likely occur, that has been shown to occur, and will perpetuate this response. So to summarize these metabolic functions of the microbiome, and this is studies that I've shown as well as others. Basically, the microbiome contributes to immune function, nutrient formation, detoxification. Also regulates food fat storage, appetite control, extracts energy from carbohydrates and proteins, identifies dangerous bacteria and neutralizes them, As well as nutritionists, we all know it synthesizes B vitamins, vitamin K. These are very good reviews that I recommend to read. Continuing on with bacterial influence on metabolism and obesity, these are the experimental results. Exciting studies that are going on now. And the first one is that obesity alters gut microbial ecology. Basically, they showed changes in the flora, and I have to give you facts based upon the fact that I've talked about humans have the two largest phyla, the Bacteroidetes and Firmicutes. These are also present in the mice, so these are the model systems that have been used for these studies. These flora, distribution changes in response to diet, and obesity of the host. The experimental model is that they use lean mice, as identified by the wild type ++. They are dominant homozygous. They also use the OB mouse that has mutation on the OB gene. They do not produce leptin, thus they do not have appetite control, and they eat out of control. These are homozygous recessive OB. They took these animals and they determined the phyla of the gut flora of the following mice. Lean homozygous, as I showed you in the slide, the thin ones. If the animal was heterozygous, but their phenotype was also lean, they were the second group. The third group, of course, were the obese homozygous mice, OB. Results are shown here as bar graphs. Both of these animals were lean, and you can see that on the y-axis we have the total sequencing of the bacterial flora. The dark bars represent Firmicutes. The open bars are Bacteroides. There's no significant difference as long as the animals are basically lean. However, the obese mice show a significantly increase in the Firmicutes and a significant decrease in the Bacteroidetes. So we have a tendency towards going to the Firmicutes in these animals. They next did this similar study with human gut flora and obesity experiments. They studied 12 obese human subjects. They were put on a low-calorie diet for 1 year. Some of these individuals were on a fat-restricted diet. The others were on a carbohydrate-restricted diet. They studied the microbiota, as with the mice, for 1 year using gene sequencing. And they again showed a change in the Firmicutes and the Bacteroidetes distribution. They, of course, showed a change in weight, being on a low-calorie diet. Here's the data. On the x-axis, let me point out first as a function of, this is at the point of when the diet was started. The blue bars represent the sequencing as identified on the y-axis of Firmicutes. The red represents the quantities of Bacteroidetes. And you'll notice as these individuals lost weight, which they did at week 52, their distribution matches that of a lean individual. And of course, Bacteroidetes increased as a function of the weight loss, as you can see on this. The next graph is the same data plotted in different ways. And the x-axis is the change in body weight, and the y-axis is the change in the Bacteroidetes, only the red one that we're talking about. The green dots represent those individuals that were on the carbohydrate-restricted diet. You can see that they lost weight, which was linear with respect to the change or the increase in Bacteroidetes. Those individuals on a fat-restricted diet also had an increase that was linear with the increase in bacteroidetes. So exciting information that the redistribution as the individual is losing weight. Taking this further and summarizing this, all subjects, no difference in the diet, had an increase of all bacteroidetes and decline in firmicutes. The firmicutes and the bacteroidetes, they dominated with, again, more firmicutes than bacteroidetes in the obese subjects. So summarizing the changes in the gut flora, obesity changes the relative proportions of divisions of the flora. Obese mice and obese people have more firmicutes than bacteroides. Dieting and weight gain or loss shifts these proportions. Conclude. Indications of the change in the gut flora certainly under these circumstances affects the obesity. Now, let me take germ-free mice, gut flora, and obesity experiments. The protocol is basically they took germ-free mice and they kept them germ-free. These are the control mice, GF. They were changed from a low-fat diet to a Western diet. Low-fat being 5%, a Western diet, 5% lipids, Western diet being 41% lipids, increasing carbohydrates. Basically, these mice were your control mice, and there were another group of mice that were given the normal gut flora. These are termed conventionalized mice. They also had the same diet change. So in this protocol, we have 4 groups of animals. 2 of them on 2 types of diets, no microbiota, and then the other groups had normal gut flora, not from obese mice. They studied the change in weight. In this graph, we can see that once the animals, these are the control mice, germ-free, they are on a Western diet. You can see that weeks after they started the diet, they indeed did have some increase in weight. Change in body weight, they basically gained 2 grams. Remarkably, if there was gut microbiota present and the mice were on the Western diet, they had a statistically significant increase in weight. From over an 8-week period, they compared to their no gut flora, they gained basically 5 grams, statistically significant. These same mice, they assessed locomotion, in other words, activity in these mice. And this is very exciting. These are represented as bar graphs against activity. And the open circles basically are our control animals. The control animals, they have no microbiota. On the low-fat chow, they had high activity, as indicated by this open bar. If they were also no gut flora, but they were on the Western diet, they were still fairly active. Once these animals obtained microbiota, even on the low-fat diet, they dropped activity. Similarly, on the Western diet, they dropped activity. So, what does this tell us? First of all, germ-free mice are reasonably resistant to the Western diet, as indicated by less weight gain, less change in locomotion. Gut flora, once they were infused with gut flora, they're responsible for increasing the energy production from food. This can explain why clients will say, I don't eat, but I still gain weight. Similarly, as exemplified by the locomotor activity, gut flora appear to influence the mouse activity. This would explain the presence of gut flora, might explain the couch potato phenomenon for individuals. So, one more experiment, bear with me, relative to the wild-type mouse and the obese mouse. They transplanted the microbiome into these individuals. The protocol was that there were germ-free mice. They were given the gut flora from either the OB mice, the obese mice, or the wild-type, lean-type mice. Chow consumption and exercise was the same, so that was not a variable in these experiments. Both sets had similar starting weight and percent body fat. And these are the results, showing that in bars, again, that the animal that had germ-free mice, they transferred the normal gut flora as represented by the ++, or they represented the OB. An increase in body fat is on the y-axis. We can see a statistically significant increase in the weight due to the gut flora from the obese mice. So this indicates that yes, microbiota certainly affect the presence of the obesity. So let me summarize this. This is work that is done by these individuals. Basically, health is on the left side, disease is on the right side. Pivotal points are our microbial products or activities. I've already talked about the microbiota will result in short-chain fatty acid production, vitamin synthesis, will influence energy supply, gut hormones, satiety, and energy expenditure. I did not talk much about lipopolysaccharide, but if this is healthy, there will be health. Supply of nutrients and energy as far as the microbiota supplying this is concerned. If this goes awry, there will be obesity and metabolic syndrome occurring. There are many studies going on talking about this microbial products either preventing cancer or promoting cancer. Next, inhibition of pathogens or source of pathogens. And we've certainly seen that with the transplants of the fecal transplant in the situation of Clostridium difficile. Next, there will be changes in gastrointestinal function as a function of the microbial products. Similarly, IBD, IBS, and of course, cardiovascular health, or in this case, cardiovascular disease. So as represented by this review article, this is a balance that is pivotal around the microbiota. So, finishing up, taking all of this data, now we want to alter the microbiome for the benefit of better health. And what are our options? We've talked about gut, disease, and obesity. We can manipulate the flora, as indicated by the experiments I showed you, through a high-fat, low-carbohydrate diet or a low-fat, high-carbohydrate diet. We can, of course, use probiotics, which has been used for years, and, of course, prebiotics that can be certainly used in these experiments for individuals for health. Next, we can certainly use stool transplants. And as I've already mentioned, there have been many studies that show that Clostridium difficile infection is overcome with a transplant from a healthy individual. And of course, this begs the question, can there be weight loss with a thin person's feces? So, going on with this, let's talk about probiotics. Basically, they are live microorganisms which, when ingested in adequate amounts, confer a health benefit on the host. The majority of probiotics are gram-positive lactic acid producers. Bifidobacteria species, which I showed you is present in the infant, as well as the lactobacillus species. Of course, they must survive the transit through the stomach and the duodenum. Others include non-pathogenic streptococci, Enterococci E. coli species, and Saccharomyces boulardii, which is a yeast. Some examples of probiotics that are on the market: VSL number 3. Indeed, large RAND studies have shown efficacy in pouchitis and IBS. But of interest, this contains 4 species of lactobacilli that are shown here, 3 species of the bifidobacteria that are indicated, and of importance, 1 streptococcus. And this particular strain inhibits the presence of the strain that produces strep infection. So this is certainly advantageous. The next one that's on the market contains Bifidobacterium infantis. It is actually a genetically modified organism by this company. It's been patented, and large trial studies have shown that it decreases symptoms in subjects with the IBS. So more promising studies with these probiotics. Another probiotic is a yeast, Saccharomyces boulardii. Again, that has been shown in RAND trials to help prevent recurring Clostridium difficile infection that we've already talked about. Now I want to go over a little bit of prebiotics. They are ingested substances that selectively stimulate the proliferation and/or the activity of desirable bacterial populations that are present in the host intestinal tract. They usually target the bifidobacteria and the lactobacilli. And of interest, this has been known since the 1950s. Studies have been done on this. These compounds are usually non-digestible oligosaccharides that were already present, as I've talked about, in human breast milk. And the prebiotic is basically an inulin. It's a plant polymer which is mainly comprising fructose units. It's an indigestible fiber, and it produces hydrogen, carbon dioxide, and methane gas. As I showed you on that phylogenetic tree of life, this is proliferating the presence of methane gas, which is an advantageous environment for the microbiota. So this perpetuates bacterial metabolism. So the future with probiotics, what are they? Question is, begs the question, is it possible to design a food, sugar, or fat that would alter your gut flora to promote weight loss? Can you give a prebiotic that decreases your energy harvest of colonic bacteria? In other words, losing weight by making your gut flora less efficient at digesting your food, not digested by the body cells. Finishing up with the presentation, I wanted to identify some of the labs that you can have your individual microbiome determined at. DNA sequencing using the PCA, polymerase chain reaction, can be done at basically Biofrontiers Institute, University of Colorado. And this is the website that is shown on here. Coupled with this is a project, Human Food Project, which is doing an anthropology of microbes. It's called American Gut. More than 4,000 people have already donated and signed up for the American Gut. Basically, you need to donate $99 to the project and you will receive a home stool sampling kit. In return, you will obtain a list of the bacteria in your sample and the relative abundance, and this will show you how your bacterial community compares to others in the study based upon your diet and your lifestyle. The funds from these studies go back to the Biofrontiers Institute, as well as it goes to the Earth Microbiome Project. And here's a website for that particular study. So now we can get into the exciting conclusions and the possibilities of the future studies with these. First of all, our gut flora influence our metabolism, weight, activity, immunity, health, and disease. And it does this by way of their genes, their byproducts, as well as their metabolic activity. Manipulating the gut flora may be an integral part of weight loss programs and different disease treatments in the future. It certainly begs the question, which comes first? Are we going for causal or correlation in some of these studies? Future, what do we need to do? Basically, there needs to be the elucidation of the gut flora in healthy subjects and those with disease. There's a study at the Scripps and UC San Diego in subjects with diet change, NASH, which is a clinical research network, bringing in many people, as well as the inflammatory bowel disease subjects. Of course, it has to be determined the gut flora-host interaction, how we respond to changes in our gut flora. And of course, designing trials that assess the potential for the probiotics and the prebiotics. All of these are very important. I wanted at this point to thank you very much for your time for me to present this information. I would love to talk to you about this information, get some ideas from where all of you are at. So here's my email address, so please contact me if you have any questions, comments relative to this information. And at this point in time, I'd like to take questions from the audience.
For the owl, right?
Yes. I couldn't resist this one, right?
Yeah, it's beautiful. The owl knows all, so we'll go with some questions here.
Really?
Thank you very much. This is a fine example of a strong academic presentation, as would be expected from educational director, and a lot of good information here. So,
Paula, it's exciting information, and I didn't go over so many exciting things. But before you get into your questions, in fact, there's a publication by Dr. Gordon, whose material I presented. He just published in Science. And maybe one of your questions is going to be, can you transplant the microbiota from a thin person into a fat person and see a change? I did not present those studies, but those results have been published in Science, and he discusses that actually on NPR on a podcast. So I had to add that because I didn't get the information in time to put it together on a slide.
all right. There was a request to see the website for the BioFrontiers Colorado University of Colorado Boulder. Can we see that slide, please, so that
yes, that's no problem
that.
at all. That's exciting.
And
Somebody
back to the owl.
Right. I know. Okay, and this is DNA sequencing that they can do. I've got that website, and then on my next slide is the, I believe it's at the bottom. I'll go on to the next one. All right, you can probably just Google BioFrontiers Colorado.
Yeah. We've got
that. But then
yeah.
here's the website for the Human Food Project,
okay.
which will be the extensive one. I have not done this. I will probably do it shortly, but it looks very interesting.
Got to try it.
I was just going to say, sometimes do I really want to know?
Well, you can go back to the owl because we can let the owl know. All right.
Cool.
So, some questions about the mice and what they were actually eating. So for the study subjects that were on the carb and fat restricted diets, do you know what they actually did eat, and if it was monitored, what the quality of the food was? Was it real food?
Right. You've got an excellent question. And again, this recent study in Science addresses that.
It comes back into the specifics of,
what it
they
represents.
didn't eat, what kind of fat, was it low fat,
Right.
low
It was
fat, what kind of carbs, were they fed kibble, was it cooked, denatured, low quality,
right.
Things like
Right.
that.
And the specifics basically are that the studies that I showed you is they represented the rat chow diet, which was basically 5% lipids, which was a normal diet for mice, excuse me, not rats. When they switched them over to the Western diet, they did not give an actual distribution of the types of fats, but the carbohydrates themselves. In a Western diet, they specifically indicated refined sugars, refined carbohydrates. So that was definitely a factor relative to it. And coupled with that, again, is this study that just came out in Science. They actually took mice and gave them microbiota. They took germ-free mice and they gave them microbiota from obese mice as well as from lean mice. And if the animals were housed together and fed what he called basically a healthy diet, which was basically low fat and high content of vegetables and fruit, then the microbiota from the lean rats basically migrated over into that of the microbiota of the obese implanted mice, and they cohabitated such that it reduced the obesity increase in these individuals. So this is the most recent study on diet, which is where you're going with that question.
Oh, very interesting in terms of that some of them got real food, fruits and vegetables.
Yes. Yes, and that's the only one that I've seen. Every other one that study that I've seen have been on the mice chow, the rodent chow that is laboratory produced. But because mice are omnivores, because their phyla are analogous to ours, being primarily Firmicutes and the Bacteroidetes, I feel that they are comparable to human studies, but not perfect.
Right. Well, perhaps you went into this, but here's another question. That mice that received the obese mice microbiota, they became obese. And the question is, could that be reversed?
That's it. That's it. In other words, taking these obese mice and replacing it with the lean mice microbiota. And those are the studies that have only been done in germ-free mice. So in other words, yes, they took the microbiota from obese mice, put it into germ-free. But I have not seen studies of taking obese mice, trying to wipe out or clean out the microbiota, and then replace it with the lean microbiota. That's what you're asking, right?
Mm-hmm.
Yes, and I have not seen those studies. It certainly implies it can be done, but again, based upon the recent studies, if it is done, it has to be done with a healthy diet. And that is key, that they are not, one will not solve the problem by just changing microbiota. The individual, I believe, still has to be on a healthy diet.
So when you say, a question here, when you say you're talking about germ-free mice, you're talking about mice that don't have microbiota.
Correct.
Correct? Yeah.
They are purchased through laboratories. They're raised in bubbles, basically. They are not exposed to any outside organisms. So when you purchase them, they're very, very expensive, You purchase them as adults, you match them up with weight, and then you, by way of colonoscopy, put in the microbiota from the other mice that you have grown up. So yes, germ-free are laboratory special animals that allow these studies to be done. And there is no analogous situation in humans. But I did show results that showed that once humans were put on the low-fat diet, their distribution of phyla did change. In other words, the bacteroides, I like to look at it that in the obese individuals, you have a preponderance of firmicutes. Upon loss of weight, there will be increasing bacteroidetes present. So you want a more diverse microbiota present, and that will promote health and a lean individual. And that's been shown in human beings.
So a lot of the research that you presented is from my studies, and so you're saying now that's applicable to humans, not so much in study, but it can be translated that way. Is that correct?
I believe it can be. It's not a perfect environment, but mice indeed are omnivores as we are. They're adaptable. They require a similar distribution of carbohydrates, fats, and proteins. And because they have similar phyla to us, I believe these experiments are applicable, as well as the human experiments that show the same distribution as a function of weight loss that are parallel.
So would the same be considered accurate, Dr. Ludwig, for diet affecting the microbiota? I mean, certainly you talked about the mice and the changes there, so we can translate that for that. Yes, diet affects microbiota definitely, and would likely be for humans also, even though not all of us live in a bubble.
Definitely. Oh, you have said that's the ultimate bottom line. Do you mind if I go back in my slides to show that study? Here I am going. Bear with me as I'm going backwards. Hopefully nobody will be getting roller coaster. I have to go back quite a ways because these are the human studies that showed that in these individuals, hang on, and this is what you have mentioned, Paula, is the whole bottom line here. This is human flora and obesity as a function of weeks on these diets. Remember, these were fat restricted, and you asked me the question, what type of fat? They did not elucidate that. But here, these humans were on a fat-restricted diet and a carb-restricted diet. They all lost weight, and as they lost weight, the presence of the bacteroidetes increased and approached that of a lean individual, so that the firmicutes that was predominant at time 0 when they started these diets, it significantly decreased, and of statistical significance, the bacteroidetes increased. So the redistribution occurred. And curiously, this bottom graph shows us that on the fat-restricted diet, there is a higher loss of weight and a higher rate of increase in the bacteroidetes as compared to the carbohydrate-restricted diet. And I know we can talk about types of fats, types of carbohydrates. They don't elucidate that relative to this information. But this is exciting data that indicating that this corresponds with transferring the different microbiota into these germ-free mice and getting the same redistribution of these phyla. And from the readings that I have done, the more diverse your human flora is, then the more healthy you will be. Where in contrast, obese individuals have primarily the firmicutes. Remember, those are the Clostridial species, lactobacillus, so some of the pathogens that are present in this. Okay?
So alteration of the microbiota is possible in two ways. You can damage it or you can support it. So if somebody was using antibiotics, they would really potentially wipe it out. If somebody was using probiotics, they would contribute to some healthy strains.
Perfect.
So in doing so, depending on which you're using, it could support weight loss or weight gain.
Right, right. As long as, and coupled with this is the importance of the diet. All of these studies, I mean, these studies are primarily talking about gene sequencing, but now they're being done coupling them with what is called a Western diet. We all know the variation involved in that, but at least they are making approach to the distribution of the fat, carbohydrates, and proteins, which will be of importance, as is shown in this bottom slide, that we have the difference.
Well, that question leads to wondering if you have suggestions for a protocol that could be used in a clinical setting to help clients lose weight based on what you know today in your research.
That's a very good question. I would, assuming that they have no other underlying issues and want merely to lose weight, yes, I would go with a probiotic and put them on a calorie-restricted diet. Depending upon where the person is going, it would be distributed based upon healthy fats and healthy carbohydrates. And that would assist with them losing weight based upon dealing with it at the microbiota level as well as the diet level.
And certainly based on the individual and their, if you can get the test and see where they're at for that, it'll make a difference too.
Definitely, definitely. And coupling it with the beauty of that laboratory is that they give you a age-matched, similar lifestyle, and showing if the person is within the norm and outside of it. So that would be coupled with it too, to do that.
So certainly from our perspective, we're talking about a whole foods diet.
Definitely.
Get it as much, if you're suggesting a calorie restricted diet in this case, then certainly they need all the nutrients they can get. Right,
because it's again, as the new study shows, high vegetable, high protein, high vegetables and fruit coupled with low fat. And again, I don't want to get into the distribution of fat because they only talk about it, I presume, saturated fats, but they do emphasize high levels of vegetables and fruits in this diet to control the obese microbiota that the germ-free mice received, even though they cohabitated with the mice that had received the lean microbiota. The value of the diet that these mice received was primarily very high, if high in fruits and vegetables.
There's an interesting website just got pointed out by an attendee about, it's called the Human Food Project. The website is humanfoodproject.com/American Gut.
Right.
So you're aware of this?
That's the one that I was referring to, I believe, on the bottom one. Yeah, that's the donation of $99. But yes,
that's right.
That will put it all together. So that's great.
Yeah, they're already looking at it, saying it looks really great.
Perfect.
So
yeah.
We just have a couple minutes here before we close, but a couple other questions. This is really you talked about the brain also in this, and a lot about fecal transplanting, and so I'm wondering about brain inflammation. Well, not so much wondering, but considering it as a health issue in many chronic conditions. I'm looking forward to some kind of a transplant for this. I hope isn't fecal.
I was just to say, really, but you're talking about the brain-gut axes. Yes. And seeing if that will change and modify as a function of the microbiota. That one paper that I had come across talks about a healthy microbiota, how do I put it, dampens the effect of the stress chemicals that are released. Maybe by way of metabolism, I can't even go there, but it does say that it ameliorates the effect of the cortisol and epinephrine as they're released under stress. It's an interesting idea.
Right. One last question. Since the microbiome has been known since the time of Mishtakov, why do you think there seems to be such an increased interest in this now?
Oh, that's an exciting
area. Really a significant topic. And
oh, yeah. There's been an explosion of data, and I directly attribute that to the new technology of DNA sequencing. In other words, prior to being able to determine the phyla of a group of microorganisms, they had to culture them all. And you know how long that takes, and you know the errors involved in culturing, especially anaerobic organisms. So the fact that the Human Genome Project came along was successful. They then initiated the Human Microbiome Project, and this has facilitated it because of gene sequencing. It's merely a technological phenomenon that now these studies can be done quickly, accurately, to show these significant changes.
Excellent. Well, this has been a very fine presentation, Dr. Ludwig. It really demonstrates the high standards of academics that you hold to, and represents a doctoral program and work our students are doing here at Hawthorne. So job well done. Thank you so much. Very fine presentation.
Thank you, Paula. I appreciate the time and the opportunity to share this information. It's been a pleasure.
Well, there was a lot, like you said, and there's a number of slides, and I want to let people know that this webinar will be available on the Hawthorne website. You'll be able to find it in a couple of days under archived webinars, so you'll have access to all of the slides and the recording. And when you're there, you'll see a lot of other webinars that have been done over the years. So you'll be able to review this webinar and a lot of others too. I do want to thank you all for attending this Hawthorne University webinar. If you want more information about the programs and courses that Hawthorne offers, please visit our website, and there's a lot of detail there. And for additional information, questions about our programs or courses, our Director of Admissions, Kathy McDermott, is always available and extremely helpful. So our next webinar, I should let you know about that. I want to invite you to join us for Tuesday, November 5th, always at 4 p.m. Pacific. Hawthorne's webinars are the first and third Tuesday of the month. And Hawthorne's adjunct faculty member, Dr. Nicholas Hedberg, is going to be presenting on the subject of autoimmune disease, the infection connection. This will be part 2. And we'll be sure to let you know more details about this in the coming week. And for those of you that want to refresh yourself on part 1, you will find that in the archives under autoimmune disease, the infection connection, part 1. So this does conclude the presentation for now. Thank you again, Janet. It's been a pleasure.
Thank you.
Thank you, James Bernardinelli, for engineering and keeping everything running smoothly. And thanks everyone for sharing this educational experience with us. It's always a pleasure to be able to offer such fine information and to join with you to do so. I wish you all the best of health and hope that you'll join us for future webinars. There's going to be a survey to fill out after the webinar ends, and it really helps us to have your feedback and comments. We consider it carefully, and we appreciate you taking the time to do that. So thank you very much for those that do, and know that it's there, and your questions, your comments, your feedback, yeah, we'll take a good look at that. So that's it for now. I want to let everybody know that we do open up for a pre-talk about 10 minutes before the webinar begins. So you can come there for announcements. We talk about a variety of different things and upcoming seminars and the like. So you're invited to join us for that as well. And until then, I wish you good health. Take care of yourself.